to wait, we will have a specific time allocated for Q&A. So you will see a screen that says questions, and during that time, if you can have your questions ready, I'd be happy to answer any of those. I'll try and not pause in the middle of something to address any questions, but hopefully we'll get through this. In the future, if you have any questions or you would like us to address a specific topic or an issue, instead of the topic popping up during the session, it would be great if you could add it to the forum. I'll do a little review of where the different elements of the program are today. And once you find that forum, you'll be able to post your questions or issues that we need to address. And then I'll dedicate time in the beginning of the session each time to make sure that that issue is addressed. So today, we have our session 2. This is the last session that is going to be open to anyone who would like to explore this program. The session is focused on multiple sequence alignment. So we'll talk about how comparison of sequences could be done using a computational method called multiple sequence alignment, or MSA. What MSA does is identify how to best align sequences and find a consensus sequence. So where do they all agree, and what is the best consensus between multiple genomes? And also, what is the quality? So how do we evaluate the quality of that consensus? And then using that alignment, how can we identify relationships between groups of sequences? So before we jump into our session, I'll just give a brief overview. The program that we're in right now is called Bioinformatics for Infectious Diseases, where we speak about the application of bioinformatics to study pathogens and their interaction with the human body. And we will talk about different aspects of bioinformatics methods using several curated data sets from coronaviruses, Ebola virus, rhinovirus, tuberculosis, and others. And the program is actually divided into 2 different parts. The first part is more educational, more theoretical, where we talk about different methods. We kind of design a pipeline and run it and discuss outputs. We start thinking about questions that are appropriate to ask from different types of data sets and how to get a conclusive answer, and what does that entail in terms of both statistical significance and biological interpretation. And then the second part of the program is more practical and more open-ended and designed to allow you to develop a project of your own. So in the second part of the program that will start in June, we will actually provide you with tools and support to design and analyze a project of your own, make sure that you get it to an interpretable result, and then give you an opportunity to present your project. And we can discuss how that project compares to other publications and projects out there, so that you get some feedback on what you've done. So that's the program that we are talking about right now. It started on May 4th, but this is only the second session. So anyone can still join this week to participate in the rest of the program. In the program, in the first part of the program, we will have several educational sessions like today, where we'll talk about different elements of viral and bacterial biology. We'll talk about zoonotic spillover. We'll talk about vectors and hosts, viral evolution, replication, bacterial infections. We'll talk about how to analyze data from viral genomes and next generation sequencing data, and what types of comparisons and analyses we can do with some of the tools that we will discuss in the context of this program. And we also prepared a number of curated data sets that are organized in a way that they show how to go from general or exploratory analysis to developing a hypothesis, and then eventually developing a full analysis with a biological interpretation in mind. And those curated data sets, we hope, will either provide you a context for a project that you will design on your own. So essentially, you can pick one of these curated data sets and take the analysis into your own direction, or provide you with a template that you can apply to any other type of a viral or bacterial project that you would like to try and work on for the second part of this program. So in the context of the program, we will provide you with tools and support for how to use those tools, as well as some educational materials that are available for asynchronous learning. So in other words, you will be able to learn about the things that you don't know using these online materials. We hope that in sessions like today, we can go through the material in a way that it helps us identify which areas are unclear, which topics need to be covered in a more comprehensive way. And we can either point you to the right direction using research articles or some of the tutorial style materials that we've prepared that combine some theoretical as well as practical exercises and go through the steps with a lot of explanation. So the program, we have a group of members that join a program, and this program is hosted on our educational portal. So this educational portal provides us with specific tools that help guide every participant to get to a place where they're more independent and familiar with the tools and the data sets and the projects and are able to conduct the analysis in an effective way. And in fact, we use some of these tools. I'll show here in a few minutes how we use those tools to interact with the participants in between sessions, making sure that everybody stays on the same page and is able to continue without getting lost as we go through a lot of these different topics. So let me briefly show you what I'm talking about. This is the link. I'm going to put it in chat. For all of you that are already participants, you should be able to, thank you, Bipsha. You should be able to find that fairly easily. And then those of you that are not yet participants, you'll be able to see what it looks like once you join. So this is the page. You will see here a few different tabs: home, forum, courses, project, progress, assignments. And then here you can open a little more and see here a few other menu items. Not all of them are going to be visible for you if you're not logged in. So the first thing that you need to do is register on this portal and make sure that you have a completed profile. Completed profile is important not just because we want to see your picture and we want to know what's your name. It's also important if you want to use the certificates that you get at the end of the program. They're generated automatically and they actually use your name. So if your name is something that is not going to look like cool boy 123, right? That's what's going to appear on your certificate. So make sure that you're updating your profiles as you join the program. Now, as I mentioned, we have a forum, and on this forum, we interact around specific topics. So for example, we post here previous recordings, and you can see that Bipsha already started posting our previous recordings from the session that we had last week and that we had this week on Monday. So if you want to see those, you can go in here and you'll be able to see the links on YouTube. And if you have any questions about those specific sessions, you can post them here as well. We all get notifications. Sorry, we all get notifications about your questions. Also, I opened a topic on the forum for specific questions. So right now, there may be not many questions as everybody is just getting oriented. But as you have specific questions, maybe a pipeline doesn't work or some other issue that comes up, please post it right here. And so I'll have enough time to prepare for the next session and make sure that we have everything ready. Here we also have session data files and presentations. So starting from this session, we started posting here a PDF of the slides. So a lot of you requested the slides from last session. We will post that here as well. But for this session, if you would like to have the presentation, the PDF of the presentation, and the data files that we will use today. So all of that is organized right here. Okay, the other thing that we talked about is that we actually monitor activity of the group that is participating in the course, and we try to understand that you're doing the right thing. So I just want to point out how that happens. So here you can see the different participants of the program and the different activities. For example, somebody ran a pipeline or somebody completed a course. So we discuss the outputs and the issues that we see. For example, someone might run a pipeline that really isn't going to produce the type of output that they're expecting. And so we will, discuss those issues as they are being worked on. So these are the educational kind of features that we've designed into this program that help us make sure that we understand where the issues are and address them in a timely way. So let's take a look at an overview of where everybody is. This is just for those that have already registered and have completed their profiles. You will see a general overview of this group in terms of the progress. And by the progress, we mean practical activities, running pipelines, as well as completing coursework. So you see that we met the first time on May 4th, and we discussed next generation sequencing, what types of sequencing methods there are, and how those are applied to sequence viral genomes and host transcriptomes. And then today we are talking about multiple sequence alignment and phylogenetic relationships. So you can see those updated on this chart. And these two graphs show us the activity of the whole group in terms of completing online materials, as well as running pipelines. So the gray is the pipelines and the yellow are the educational activities. And here you can see some of the participants and what they are working on in terms of practical or educational components of the program. So this is just a good overview of where we are as a group. Okay, so we talked about the forum. We talked about the sessions. If any of you want to see previous sessions, you can go to the forum and check those out. Beepsha, maybe you can post the link to the forum just so that everyone can find it. We also talked about the progress and today's session. And so now let's review what we did in our last session. So I saw some of you try to replicate some of the pipelines that we did, and maybe had some challenges. And so let's just make sure that all of that is clear. So last session, we talked about next generation sequencing. Next generation sequencing refers to the second generation sequencers like Illumina sequencers. The second generation, the first generation sequencers were the Sanger sequencers, and these are the second generation. And of course, to generalize that, we can call that next generation. And the difference between the first generation and the second generation is that the second generation is called shotgun sequencing. It sequences random pieces from a collection of shared material, DNA or RNA material, and produces short reads. And one of the bigger challenges in then understanding where those reads map to on the reference genome is called alignment or mapping. And so last time we talked about how we can take sequence read archives that collect an archive of reads, of short reads, and map them onto a database of different available genomic sequences from viruses, bacteria, and other organisms, and then also map the reads that we identify belonging to a specific organism on the reference genome of that organism. So what you see here are some of those reads aligned to the reference genome of Ebola virus. We talked about different approaches to metagenomic and viral sequences. So we talked about how we can design specific primers to identify a virus or a group of viruses like a species. And those will be designed specifically for a conserved region or a gene. For example, we took a amplification of the Ebola virus specific gene that can help us identify presence of Ebola virus in fecal samples from bats. There are other approaches as well. For example, we can do no amplification and just do total DNA or RNA. A good example that we started talking about last time is a project that is available for everyone called Spotiptera frugiperta, which is a cell line that uses a plant cell line to produce different biologics, so vaccines and proteins that we use. So there we saw how you can sequence the expression, gene expression data from that Spotiptera frugiperta, and then take the not mapped reads and then align them to, again, the same database and identify presence of very specific contaminants. And then also there's what's called environmental microbiome metagenomic sequencing. That is actually designed to help us identify diversity in a given environmental sample. Environmental could mean taken directly from the gut, or taken from feces, or taken from water, or taken from soil. And so what we can see there is really trying to look at hypervariable regions that all come from, for example, a bacterial 16S ribosomal RNA, and help us understand diversity and characterize the community of different bacteria. So we talked about those three types of sequencing methods. We also ran a pipeline, like I mentioned, and so I just want to make sure that we all understand what that pipeline was and what was it supposed to produce. As I was seeing what people were running, not everyone got it right the first time. So just to remind everyone what it was that we actually did. So what we did was we took an SRA file, a sequence read archive. So this is the name of the archive, SRR 107-69500, which is a metagenomic amplicon of Ebola virus, but specific to a bat feces sample. And so that file looks like this. So you can see the sequence read archive name right here, and you can see those short reads, their sequence, as well as the quality of that read. So we took this file, it's a pretty big file, hundreds of megabytes, and then we uploaded that file and used it to map on the virus genome. So we have a database from viruses of different viral genomes from NCBI, and we just used bowtie to align, which is a method of alignment of those reads onto that database. And then we also created an abundance table, and that is a script that allows us to calculate how many reads fall onto one of those genomic elements. So as we did that, we had an output. One of the outputs was a table, and that table showed us that we have multiple different accession numbers, and those accession numbers contain, if we look them up on NCBI, one of them would be Ebola. So there's influenza, there's other phages, bacteriophages, but we were interested in Ebola. And so then we ran a second pipeline, and that second pipeline took the reads that we identified that fall onto that reference genome, and we actually produced a visualization. And that visualization looked like this, where we saw that there are reads that aligned in a certain position on this Ebola virus genome. And that is kind of where we ended, right? So we saw the reads, how they align. We talked about coverage. And then you probably noticed that some positions were different from the reference genome. So for example, here it's C, here it's T, right? So those are the differences. And this is what we are going to be talking about today. So today we're actually going to be talking about how do we go to the next step and understand what are the differences in the sequences that we are finding. Okay, and here's just a description of how that protocol for amplification of different types of viruses. It's actually not designed specifically for Ebola. It was designed for 16 different viruses, including influenza, Ebola, and other viruses. And so that's why you see here others present as well. Okay, so before we go on to today's session, I want to make sure that we have addressed any of the questions that you had about sequencing techniques and how the first step of once I generated some sequences, how do I know what sequences these are? So if you have any questions, again, put them into chat. And let's talk about multiple sequence alignment next. Okay, so if there are no questions, let's start talking about multiple sequence alignment. So multiple sequence alignment will deal with variation in sequences. And so the variation in sequences, as you can see displayed right here, has to do with changes in one sequence compared to another. So we're not going to deal as much with how different reads map and where they map, but more how different individual nucleotides vary between different sequences. So where do these changes come from? We are talking about viruses right now. So what we are talking biologically is taking samples from cells and looking at the presence of different species that we can observe in a sample, or the different variants of a virus. So for example, when a number of viruses enter an organism or attempt to enter an organism, only one of those might be successful. So again, there are many viruses in our bodies at a given moment in time. Those viruses, the majority of those viruses are actually helpful, if not harmful. They're helpful because the majority of the viruses affect bacteria, and we need some balance between bacteria in our intestines, for example, and in different places of our bodies. And so viruses are actually a part of that ecosystem. But some viruses are able to enter cells. And so when they enter into the cells, their goal is to go through the steps of viral replication, imitating a sequence that would be native to the cell. So they are imitating, trying to get the cell to incorporate that into its assembly line. And that assembly line will help produce proteins that then replicate other elements of the viral sequence, of the viral genome, and put together a whole virion that then eventually will be expelled from the cell, and oftentimes the cell will die. Because the cell dies, that causes inflammation and immune response. And so there are many different areas in this process that are pressures on the virus, right? So the virus protects what's inside, protects the genome. Typically that genome is either a DNA or an RNA. It's inside a virion, and once it's inside the virion, nothing happens. It's static. But once it gets out of the virion during the process of replication, different changes start happening to that sequence. For example, the reading of the sequence to produce different elements and reproduce other copies of the same genome are going to cause different changes. Sometimes when this community of viruses is expelled, only specific virions will remain intact to infect other cells. And so that process of going between tissues, entry into the cell, replication, all of those cause different mutations or changes to that genome. So as a result, we have a community of different viral organisms or viral genomes, and they accumulate mutations in their genomes. So how can we study that community? We can study that community by studying those changes in the genome. That is a process of viral adaptation, and we know that it has a specific mutation rate. And it's actually a remarkable capacity of these viruses to adapt to new hosts. So, for example, we talk about viruses going between bats and humans, or or human primates, non-human primates, right? Monkeys, and uh, those changes are occurring because of accumulation of mutations in key regions of the viral genome. And so the rates of spontaneous mutation vary between different viruses. RNA viruses, for example, mutate faster than DNA viruses. Single-stranded viruses mutate faster than double-strand viruses. And genome size appears to correlate negatively with mutation rate. So the longer the genome, the less mutation rate. Viral mutation rates are modulated at different levels, including polymerase fidelity, sequence context, template secondary structure, cellular microenvironment, so where exactly do they replicate in the cell, replication mechanisms, proofreading. Some viruses have a proofreading mechanism to correct any big changes, and access to post-replicative repair. And massive numbers of mutations can be introduced by some virus-encoded diversity-generated elements, as well as by host-encoded cytidine adenine deaminases. So the cell has specific ways to make sure that deleterious mutations occur in the viral genome. They make sure that when they identify this to be a non-human, for example, genome, there are specific mechanisms to intervene there. And of course, the virus has to evade all of these different responses. And so this is a process of virus host circuitry that exchange different responses and different mutations. As a result, we observe how these sequences change over time. And this is a response to this ongoing process. Now, as we talked about, different viruses have different genomes and different structures. So, for example, here you can see mutations per site per year. This is the number of mutations per site per year, and this is the genome size. So here you can see single-strand DNA viruses, double-strand DNA viruses, RNA viruses are right here, bacteria, eukaryotes, etc. And so we are way down there, because mutations are actually a natural process, including for mammals as well. So to study how these different mutations characterize different strains of viruses, even within one species, for example, the Ebola virus, what we want to do is to identify how the present day observed sequences of genomes could be explained by phylogeny. In other words, how did they originate from a common ancestor? So what we have is the concept of evolutionary time on the x-axis, and we have similarity on the y-axis. And so what we are trying to combine is a way to represent some kind of an organizational structure, taxonomic structure, between the different species that we have occurring now and suspected origins that we know should have existed in the past. Now, there are many different visual formats to actually visualize phylogenetic trees. This is a phylogenetic tree, this is a phylogenetic tree, and this is a phylogenetic tree, so they all show the same thing. And what we'll talk about today is how to generate a phylogenetic tree based on multiple sequence alignment, and then how to visualize it, and what do we see when we visualize that kind of a tree. To do that, we will use a project that we already started working on, Ebola, that has very specific mutations that were identified in the context of the 2014 to 2016 epidemic. And this is based on a publication, the evolution of Ebola virus, insights from the 2013-2016 epidemic, that was published in Nature Review in 2016. So what happened with this outbreak? Ebola virus was first detected in the 1980s. So the human sequence of the Ebola virus was detected back then. Back then, it was sampled from Gabon and Zaire. And eventually, so you can see here, this is a map from National Geographic. You can see in 1976, there were 318 cases and 284 cases in Sudan. And the first outbreak was in 1976 in Sudan and Zaire. And in Gabon, there were some outbreaks as well. But this novel outbreak in 2013, 2016 happened right here in this region of West Africa, in Guinea, Sierra Leone, and Liberia. And you can see here how there are different kind of epicenters for this outbreak. The suspected source for this outbreak, again, was zoonotic spillover. In other words, the virus came from some kind of a bat, perhaps, to a primate, and then eventually to a human. So Ebola virus is the causative agent of Ebola hemorrhagic fever, EHF, a disease that affects humans and other primates as well. And the incubation period for EHF is 2 to 21 days, and typical early symptoms include fever, chills, and other symptoms that cause multi-organ stress and subsequent failure. So in other words, the organs just stop working, and blood starts coming out of everywhere. There's very high death rate, as high as 90%. And worse yet, it is highly contagious, spreading through direct contact with infected body fluids or skin or mucous membrane contact. And so this is something that has been a big concern for international organizations, of course, to contain this disease in a small, numbers of people and location. But in 2014, the epidemic in West Africa, of course, was a big deal. But as a result of that, we had the opportunity to study Ebola virus and understand how Ebola virus goes between different hosts, goes between different hosts like bats, apes, and humans, and also understand how it spreads. What is the epidemiological aspects of Ebola virus? So that's what we'll talk a little bit about today. Ebola virus is also related to other similar viruses like the Marburg virus, and is the member of the family Phylogeny, and it's named for their filamentous shape. So this is what it looks like, kind of like a worm. It's an enveloped, non-segmented, negative-stranded RNA virus. Ebola virus particles have at their core a viral nucleocapsid, which is these green things, nucleoprotein, nucleocapsid, composed of a helical single-stranded RNA genome wrapped around viral proteins NP, VP35, VP30, and L. The nucleocapsid is surrounded by an outer viral envelope studded with a viral glycoprotein spikes, GP. So we'll talk about glycoproteins in the context of other viruses as well. And viral proteins VP40 and VP24 sit between the nucleocapsid and the envelope. The viral cycle begins with host cell entry through a poorly understood mechanism. Once inside, the viral RNA-dependent RNA polymerase binds to the approximately 18 to 19,000 kilobase genome as a complex with other factors, and transcribes the negative strand genome into a positive strand mRNA. So the goal of this virus is to imitate, like we said, what happens naturally in the cell. And so it tries to show the cell this negative strand RNA so that the cell will think that it's an mRNA ready for production, so it makes a positive strand mRNA. And what's important to understand is that this whole genome has a specific organizational structure for these different proteins that you see here. So what do we see right here? And we have a question here, how to make a phylogeny with molecular clock. And so let's talk about what we see on this side. So here we see different strains identified of Ebola virus This is again the accession number. We started talking about accession numbers last time. Here you can see where they were taken from. So Gabon, Cote d'Ivoire, Gabon, and Guéquigidou, which is actually somewhere on the border between Congo and Gabon and Liberia. So what we are seeing are different strains and the dates where they were, when they were sampled. And this is the 2014, so one of the first sequences that were actually taken during that outbreak from 2013 to the 2014. 16, sorry, is the red line the reference strain? No, this is actually that new sequence that caused that epidemic in 2013, 2016, right? So the question was, one of the original questions would be, where did this originate from? And how is it different from previously known and studied viruses, right? So we know a lot about these that occurred in 1970s and 1990s and early 2000s. How can we know what is different about this one? So that might give us insight into how it will replicate, how it will spread in the population. Okay, so how do we then interpret this phylogenetic tree? So the phylogenetic tree, as we said, seeks to show us distance in terms of evolutionary time and link that evolutionary time to real time. Since we know real time of sample collection, how do we understand how they are related? And so if they were related, if they were closely related, we would see that this specific species would be somehow connected into one of these other known strains, right? And then it would give us a better understanding of whether it's something completely novel or something that we already dealt with before. How do we do this kind of analysis? Okay, that's the next question. How do we make these kinds of trees? How do we align those sequences? How do we actually study this kind of data? Let's talk about that. So the first step is to do an alignment, right? So alignment is a fairly simple task. It does not require any kind of complex software. In fact, I will show you today how you can do it in a matter of minutes, all of us together on NCBI. So freely used software, you don't need anything else, based on assembled genomes. To do that, let's go to NCBI virus and select a certain number of sequences. Okay, just before I switch over to NCBI, I'm going to give you those specific sequences that we would like to select. Okay, and these are going to be accession numbers. Okay, so let's go to NCBI virus together, and we will do this together. And for those of you that would like to just get to the page that's already prepared and not go through the steps together with me, I am going to just also paste here in chat. Okay, I'm going to paste in chat this link so that anyone can just directly go to that page. Okay, maybe keep it open. Okay, so let's go to NCBI virus. NCBI virus, sorry, to right here. So NCBI virus, to get there, you just go, yes, if you want to follow along, please follow along. That will give you some, independent practice. And next time, by the way, we have a whole session dedicated to running pipelines together. So we will actually do a very detailed review of today's and run some pipelines that are meaningful. Okay, so search by virus. Here we can go to Ebola. Okay, you can see Ebola virus come up right here. And now we have over 3,000 different nucleotide sequences. Of course, there's a lot of different things here. Not all the hosts are interesting to us. So what do we want to do? We're actually going to filter this by accession. So what I'm going to do is take that list of accession numbers. I'm going to paste them here. And I will find those accession numbers right here. So there's a total of 9 different sequences. Where are they from? There are some from Liberia, 3 from Liberia, 3 from Gabon, and 3 from United Kingdom. Okay, why are we selecting these specific ones? All of them are from human, but the reason is because we have an older series of sequences, the original ones from Gabon from 1996, not original, but somewhat old, 1996, and then a couple or six between the different locations. They're essentially the same time, so all of them are 2014, 2015, but some are in Africa and some are in Great Britain, in the UK. Okay, so we selected them just to kind of get a sense of the logic, right? We are looking here at different genomic sequences. If I click on this accession, I will actually be able to go there and see what that accession looks like. So I have here all of the sequences in amino acids. These are amino acid sequences of independent genes for different proteins. And then if I go to the bottom, I have the nucleotide sequence in TCGA, right, for the full genome. And you can see that the full genome is just under 19,000 nucleotides long, okay? So what we'll do first to understand how multiple sequence alignment is, I'm just going to select all of these 9, and I'm going to align them. So just click here on align. You'll have to wait just a couple of minutes before the alignment is done. So as I said, it's not a very complicated task, but here you can see the alignment. Okay, maybe it's not a very comfortable way to view the alignment, but it kind of makes sense, right? What are we seeing here? We see here some kind of a reference or a consensus, and we see the individual sequences right here. And what you see in red, if you go to coloring, you will see that we are looking at show differences, and we can look at frequency-based differences. Okay, so even NCBI has some issues, but what we can see are some differences, right? So differences between them. So what do we see here? We see 3 sequences that have a lot of differences, and then we see the rest that only have some references or some differences, right? So why are they so different, right? So the answer might seem obvious. The answer is probably because if we hover over here, we see what sequence this is. Okay, for some reason, it's not showing everything, but we can see that this is Liberia. Again, this is going to be Liberia. So all of these, you can go through them. All of these top ones are the novel, the new ones, the 2014. And these bottom ones are the ones from 1996, right? So we can see just based on changes in the nucleotide sequence that there are changes, a lot of changes between these 3 and the other 6, okay? But how do we now visualize their relationship? How are they related to each other? So the other option that we can do is to go back here, and instead of alignment, build a phylogenetic tree. So we're going to click here. And again, fairly quickly, it is going to show us those relationships that we're interested in. So for example, here you can see United Kingdom, Liberia, United Kingdom, Liberia, Liberia. So all of these are the 2014 and 15. By the way, the 15 stands out. And these are all the 2014s. And here on the bottom, you have the 1996. Okay? So this shows us, this phylogenetic tree shows us whether they came from some kind of a common origin. So indeed they did at some point, but realistically they're very different, right? So these probably came from a common origin a long time ago. Here we don't see how long ago, but a long time ago. And here they're really not interrelated. Okay, so this is just a very quick overview of what you can do to understand some of these sequences. Now we can download these sequences. So here I downloaded, and we can look at a more detailed characteristic of each sequence. And what we'll see is that some of them have been directly sequenced from the wild type, for example, from a patient sample from their blood. Some of them have been tissue cultured. So in other words, they were taken from the blood, then put into a tissue culture to amplify, generate more virus. And then they were sequenced, and they come from different country, collected in different year, and they belong to different strains. And those different strains are, visibly different here as well. And then this is how we will group them in our further analysis. Okay, so what are some additional considerations to this simple analysis that we quickly did on NCBI? Well, we have to think about, what do these nucleotides represent? So again, like we said, the virus is trying to get into the human cell and imitate what happens inside the human cell. So what happens inside the human cell? Inside the human cell, the cell has the DNA, and the DNA is organized into a double strand of these different nucleotides connected together into what we call base pairs. And tricodon, so the codons are trinucleotide sequences that form different types of amino acids, right? And that's what this structure actually defines what these different genes encode for, right? So these genes in the DNA are, whenever the cell wants to produce a certain protein, it opens up the double strand in a specific position of the gene start, transcribes that into mRNA. So it produces a complementary mRNA to the DNA, and then translates that sequence of mRNA into protein. And protein is made up of amino acids that fold in a very specific way. So to understand how this virus changes and what are the different changes that characterize what we just saw, that relationship, we need to understand the impact of those substitutions, deletions, or insertions by what do they represent from a protein standpoint. So to understand that, let's think about the different types of point mutations. So different types of point mutations could be of three different kinds. They could be substitutions, where one letter is substituted by another. Insertion, when a letter is inserted as that process happens, or deleted. And so as a result, we'll have different types of consequences to those different changes. What are those consequences? We have essentially four different types of mutations. So one of them is sense. In other words, it doesn't change anything, right? It just substitutes one letter for another without changing the amino acid. And that is what we call a neutral mutation. That mutation doesn't change anything in terms of what it represents. But then there are three types that actually do make a big change: a missense mutation, a nonsense mutation, and a frame shift mutation. So a missense mutation is where there's a change in the amino acid, right? And so the change is also important. Some amino acids have different physicochemical properties, and so the bigger that change from one property to another property, the bigger the impact on the protein that it produces. Nonsense mutations, they create a stop codon that terminates translation, right? So in other words, you can get a shorter sequence. So just by changing specific nucleotides, you will get a very different protein that might have a very different function. And then you have frame shift mutations. Those can be from addition or removal of additional bases. And so you can have a sequence that is completely different from the original protein, right? So this is important to keep in mind. So whenever we do this analysis, whenever we do this alignment, what we saw in NCBI is very basic. It just shows us changes at the nucleotide level and relationship based on frequency of occurring nucleotide changes. But here we want to actually look in a greater detail and understand the impact of the different changes, and then to understand how, from an evolutionary standpoint, those might actually take place and the impact that that will have on the viral sequence. So what we have to do is to make sure that we convert the nucleotide sequence by using codon mapping and understand the amino acid that is produced from these trinucleotide codons. And we have a specific chart that we can follow that is known from the different changes in the different positions of those codons that make a specific change. So we will now do a similar analysis, but in a much greater detail. So what we'll do is we'll take the nucleotide sequence, we'll convert it to an amino acid sequence alignment, and as a result, we'll be able to incorporate some evolutionary analysis into the phylogenetic tree to understand evolutionary distance, okay? So let's do that together. This is what we'll do. We'll take those same 9 different sequences that we already looked at. Those came from Liberia, Gabon, and Great Britain. And we will create a simple pipeline, do these steps. So we'll do multiple sequence alignment based on nucleotide sequences, then convert them to amino acid, and then also look at codon changes. So which position in the codon the change occurred to understand how evolutionary difficult or simple that change should be, right? And then we will create a phylogenetic tree based on a model. And so I'll talk a little bit about the different types of evolutionary analyses to build that phylogenetic tree. To do that, Bibsha was posted some of the files that we'll use to do that. So please find that link either on the forum or maybe Bibsha can post it here again for anyone that wants to download. And of course, this is going to be using the Bioinfo platform where you have already probably been able to visit and play around with. Okay, and after this, we'll have some time for questions. So let's quickly do this before our session is over. We will go to areas of analysis. Okay, so this is our server platform, and again, your pipelines are monitored there, so we can help you if you have any questions and see what went wrong. So please navigate to server t-bio.info, and here we will use phylogenetic tree. So this section right here. For those of you that don't have access, perhaps you only have access to a demo account, you can also use the demo. So there's a demo right here that you can follow along with. But those of you that are participants of the program, you can join here and we'll actually do the analysis together. Okay, so these are going to be FASTA files, right? So we're not using FASTQ files right now, we're using FASTA files. Click right here. Right here, we want to upload some of those files. So we'll just click on add files. And this is the data that Bibsha shared with us. And here we essentially have 3 different FASTA files. So those 3 different sequences for each type, for each group, are inside, right? So here we have Ebola, 3 files from UK, 3 files from Liberia, 3 files from Gabon. So what we want to do is just select all 3 of them and upload. Okay, then we want to organize them into groups. So we'll create 3 different groups and put UK, Liberia, and Gabon. Okay, we could have done this by downloading just individual FASTA files and then organizing them here as well. Okay, then we want to have some kind of an outgroup reference, right? In other words, we want to have some file that is very specific to a sampling date, right? Because we want to associate this with real time. So we're going to add here, okay, if you read the directions, upload 1 GenBank file for framing codon. So we want to have a GB or a GenBank file, which is the original sequence of Ebola from Zaire from 1997, right? Because we have a sample here from Gabon from 1996. Okay, then we have two files, GenBank or FASTA, so that we can estimate evolutionary time, so that it understands what the evolution looks like. Okay, and so we have those here from Gabon 2002 and Liberia 2014. Okay, so we add these files here. This will establish the evolutionary time. And now we're ready to start. So we start here. We want to do multiple sequence alignment, just like what we did, right? Based on nucleotide sequence. Then we convert that sequence to amino acids, and you can see that there are different charges to the amino acids. So we'll incorporate that into our alignment. And the MSA position, right? So this is specific codon positions for further phylogenetic analysis. This is something that is based on a publication. I encourage all of you to look at this publication to understand how the different codon positions have an impact on the evolution. So here we can select codon third position and 4D, for example, as true. Okay, this will take a little bit longer. If we select it as false, it will take a little bit faster. And now we have a choice of different types of phylogenetic trees. So there are different models. Essentially, a phylogenetic tree is a mathematical model that accounts for the evolutionary process and tries to match it with the data that it has available. Now, these processes are typically done using Markov chain models. And this is a statistical approach that estimates how likely a change is to occur. So there are three different options that we have for BEAST. BEAST stands for this kind of a Bayesian analysis that we are doing. And those are coalescent, Yule process, and birth-death process. So the most appropriate for this type of analysis is the birth-death process, which is a model that kind of looks at both the birth, so new replication of new viruses, and death, right? So some viruses die, some viruses are born. And so here we're going to have this model, and we can select whether we want to use amino acid or nucleotide sequence, so we can keep it nucleotide. Let's click on save and click on end. And now all of you that have the option to run this pipeline, please remember to name your pipeline some kind of a way that we understand what's happening. For example, I can say Ebola virus, Gabon, Liberia, and UK. Sorry, let's try to not use commas here. Okay, and we'll do multiple sequence alignment, phylogeny, BEAST, okay? And run this pipeline. Now, I already ran this pipeline. It does not take a very long time, but I did run it. So I want to show you what the outputs look like. Okay, so here we want to look at a number of different outputs, and you can see that as you dig deeper into the details, you will realize how important these different settings are and how insufficient it is to just use straight out of the box tools that you don't know anything about how they work. And sorry, yes, I will take a pause. We don't have that much time. So what I want to do is go through these different steps and cover a few more slides so that we are more ready to actually practice this next time when we have a full session dedicated to just running the pipeline. Okay, Claudia, so I'm going to try and complete the presentation today so that next time we can do a review and do a more slower step-by-step process, because as you will see, our next session is dedicated to selecting the files and actually running the pipeline. Okay. So what we have here are different outputs in different formats. So you can see a format called .trees. We have a format in FASTA format. We have PDF files. So let's open this PDF file and let's see what's inside. Okay, so what's inside is that phylogenetic tree that has the outgroup, that has the reference genome, and has the groups of samples that we have uploaded. So the first question that we might ask is, are the groups in different positions, right? So just like we saw in NCBI, we saw that group 2 and 3, so for us, group 2 and 3 were UK and Liberia in 2014 and 2015. And group 1 was the original Gabon sequence. So here we see those 3 sequences. So this shows us that this is done correctly. In other words, we see that they are not related directly to each other. They're fairly distant, but the ones that are more closely related, 2014, 2015, you can see that the time, evolutionary time, is much shorter, right, compared to these. Now the rest of the story is in the details, right? So here we have the annotation based on the different annotation that we prepared. And that annotation, by the way, is available in the data files if you looked on the Dropbox link. And the goal of our next few slides that I'll try and go through fairly quickly as we don't have that much time, is to understand the intricacies of this analysis and what else we can learn from here. Okay, so another important file that I would encourage you to look at is this one, fulltable.tsv, which actually will show us how the different sequences are aligned at the amino acid level and the nucleotide level. Okay, so what we wanted to understand, right, is the relationship between them based on multiple sequence alignment. So this is a very primitive multiple sequence alignment. We see here what the accession numbers are, right? So each one of these represents an accession number. And those are the different sequences that we uploaded, right? So based on these sequences, we can, for example, see nucleotide sequence in the beginning. And then down here below, if you scroll down, you will start seeing the amino acid sequence, okay? That starts from around here. So you can see how the changes at the nucleotide level affect the changes at the amino acid level. And because it's in Excel, now it's easy to extract from here. It's a table. We can use anything to extract from here the right type of a comparison that we would like to do. The other file that we have is the FASTA files. And we'll talk a lot more about FASTA and CLUSTAL files that will actually represent multiple sequence analysis. But what I wanted to do in the rest of this session is to mention, going back to what we did last time, how these FASTA files that represent a consensus actually could be analyzed in a very similar way and provide us with a lot of detail about the diverse population of viruses that we actually have in a sample. Again, I'm sorry, somebody's got their microphone on, so if you could please mute the microphone in the meantime. Okay, so as you remember last time, we aligned reads onto the reference genome. So let's review what are we actually seeing in this visualization from last time. We have the reference genome right here in the nucleotide level, and we have 3 different versions of how this nucleotide sequence converts to codon sequences or amino acid sequences. So you can see, depending on the reading frame, 3 different options, right? And so you can see how the read sequences change in specific positions, and we call those single nucleotide polymorphisms, SNPs, or variations, single nucleotide variations. And we discussed about those having different types, right? So here, for example, you can see a G instead of an A. Here you have a C instead of a T, etc. How do we actually look at those in FASTQ files? So we have a short sequence in every read, right? And together they form coverage, what we were just looking at. We can see three different types of variants here, right? So for example, here we have a very strong difference in a sense, right? Because we have 1, 2, 3, 4, 5 different reads that show us a difference from the reference genome. So we're pretty confident about the substitution. We call it a substitution. In this position, we will call it substitution 1, 2, 1, 3 from T to A. Another option is if we have a number of reads, like we have 2 reads right here, that all show a gap. And that gives us high confidence that there's an insertion or deletion. Of course, 2 reads is not a lot, but just for illustration purposes, right? And then sometimes we will have conflicting evidence. So sometimes we might have reads that actually show different substitutions. And then we actually don't know what is that substitution, right? And our confidence is low. So as a result, we can start looking at a number of things inside a FASTQ file. Okay, so we'll talk about this more next time, but one of the options that we can do is look at different consensus sequences present inside a FASTQ file. So for example, here we see how different position mutating on the genome represents different similar sequences inside a FASTQ file. Okay, and the question here, what does the blue line indicate? So these are just reads, right? So these are just reads, and those reads contain different types of mutations, right? So then we can organize them by similar mutations, mutations in the same positions or similar positions, depending on how we want to do that, and say that there's actually multiple potential genomic sequences here that could be assembled together, and we call those haplotypes. Or they're also sometimes called viral quasi-species. We'll talk a lot more about this in future sessions, but this is just for us to understand. The other option is we could just generate consensus sequences from multiple groups. For example, we had 3 groups, right? And I'll just show these 2 pipelines. We had 3 groups, and we want to understand what is a consensus in each number of files in each group, right? So for example, what is the consensus from the Gabon? What is the consensus from the Liberia? What is the consensus from the UK? And we can put in a lot of sequences in each group to generate a consensus. Another option is Shura, right? This this is an assembly of haplotypes, right? It won't look at different groups that we define. It will look for all the present haplotypes that are likely to be present in the files that we have. Okay, questions. Now let's go to questions. Is it necessary to take reference genome for making MSA and tree? MSA does not require reference genome. MSA requires at least 3 sequences to be present. So if you have 2, that's not called a multiple sequence alignment. Multiple sequence alignment starts from 3, and then it will just align them to each other. If you have a reference and 3, or one of the 3 is a reference, you can then calculate divergence from the reference. For a tree, again, you do not need to have a reference, but if you do have a reference, it is better to estimate the divergence, and that could be translated into the evolutionary time. Okay, if you have any other questions, you can put them in here. Now, next time, we will talk about a different sequence analysis section where we can combine these different analyses together. And what we'll do next time is we'll actually apply this analysis to the coronavirus data set that we will together find on NCBI. Okay, so today we talked a little bit about Ebola. We'll do that same thing for COVID-19 and SARS coronavirus and beta coronavirus and MERS. So we'll talk about that again and practice that in a little bit of a different way so that we can really understand the logic. Okay, question here, do the references always the older? Well, not necessarily, right? It depends on the question that you have. And that's where I want to end this session today, is an assignment, okay? So let's think about this. We talked a little bit about Ebola virus, and we talked a little bit about coronaviruses, and there are many public domain data sets available on both of these outbreaks. So what we want to do for our next session is to think of a question that you have, okay, so that we can organize those data sets in a meaningful way. So how do we do that? I showed you today NCBI virus, today and last time. So you go to NCBI virus, and the first question, just so that you have an opportunity to explore and see for yourself, is to understand what is the first available sequence of an assembled genome for Ebola virus from a human host. Okay, and Yala, this could be a good question for you. How do we know how good that assembly was, and how was that assembly done, and how was the sequencing done? That will give you some sense of whether you want to use that as a reference for a novel outbreak. Okay, the second question that I have for you is, how many different hosts can you identify with a full genome for all types of coronaviruses? Okay, so we talked about Ebola virus. One of the questions is, what's the first one? How do we know what was assembled and who did it, right? So let's just look into it and find it. The second one is coronaviruses. Can you go into NCBI virus and identify how many different hosts was coronavirus sampled from? Okay, coronaviruses are abundant in cows and in pets like dogs and cats and humans and bats and apes. So how many in total different hosts can you find with a known full genome? Okay, make sure that you're looking for full genomes. And let's compare what we find next time. And finally, the real assignment is try to find a bioproject. Okay, so this is a little bit more than just looking on NCBI virus, which is kind of easy to use. But now let's talk about bioproject with a sequence read archive samples, right? So they will be FASTQ files, but they will be compressed in SRA files on Ebola or SARS-1 samples from human host sequenced by Illumina. Okay, you'll find a number of them sequenced by Oxford Nanopore. You'll find some of them sequenced by other sequencers. We want to focus on Illumina sequencers. And we want to find a number of different sequence read archive samples, SRA samples, that are organized into a bioproject. And so we'll have a chance to start thinking about, okay, how do we find these different sequences, and then plan our analysis in a short session. So what is the next session that we'll have? Our next session on May 12th is going to be dedicated to the preparation and running of your pipeline. So we will review finding full genome sequences and preparing those FASTA files, selecting the right kinds of genomic sequences, and preparing a full pipeline of MSA and phylogeny to answer a question that you might have. Okay, so that will be the focus of our next session. Okay, so that's what I hope that you all can start working on for the next time. If you have some questions and you want to follow a step-by-step tutorial, first of all, you will be able to review this recording and do the same steps. The second one is what I recommended last time, to really start from what we did last time, which is mapping reads onto the reference genome, and that is covered in that SF9 project. So Beecha, if you could post the link to the SF9 project, that has a very detailed step-by-step explanation of what to do with the first pipeline. And I really was looking to see that some of you would be able to go through that project to really make sure that you understand that step-by-step sequence. Okay, and analysis with free software. So I think you got what you were looking for. I showed you the analysis with NCBI. And with NCBI and those free tools, of course, are publicly available. In terms of the tools that we're using here, they're also open source programs, right? The only thing we want to avoid here is, dealing with technical issues of installation and, making sure that it works on your computer or on my computer. So that's one part of the story that we want to avoid doing this program. As you can see, we're moving pretty fast, and our goal is to get to a research result, not, figuring out how to make your computer work with a certain program, which is typically a very time-consuming task. The other issue here is that some of these files are huge. We'll be dealing here with files that are, many gigabytes in size per sample, and we'll take many samples together. We'll, do something like 20 and 50 samples. So these pipelines will generate a lot of data, and, I'm not sure that everybody, I, for example, don't have that much space on my laptop. So unfortunately, that's just not feasible. Okay, and for Claudia, it is difficult to see your screen and simultaneously open the server web for doing the pipeline. Yes, I'm sorry, I understand that. And so that's why we'll spend next session doing it very slowly and making sure that all of you that are participating can do the exact same steps, and I'm not rushing ahead. And also, if you view this recording that's going to be posted very soon, you can pause and try to do those steps and run the pipeline, and we'll actually comment on your pipeline if it wasn't done the right way or there was some issue. You can see that we've already been interacting with a number of users that have been doing that right here on the home screen. So please go and review here. You can also link. You can actually go to some of these pipelines that other people ran and see what is going on. So, try it. And I think everybody learns by trying. And again, this recording, I think, will be a good guide to just repeat the steps. But in the future, right, when you're ready to kind of work on a project from beginning to the end, we'll have the projects available for Ebola and for coronavirus and for SF9. So take a look at those as well. Okay, so does anyone else have any more questions about what we did here today? Okay, there are no more questions. Again, I'm looking forward to seeing you again. This is the next session, preparing and running your pipeline. If you had any questions between now and then, please post it on the forum. On the forum, you'll have the recordings, you'll have the sessions, the files that we used today. Right, so right here, session data files and presentation. So you'll see the presentation for today in a PDF format. If you have any questions, please put them here. Recordings recorded right here. And if you have, anything else, you're welcome to post it there. Take a look at some of the courses. Okay, if you want a tutorial to follow step by step and run some pipelines to understand how all of this works, at your own pace, here's multiple courses that you can start looking at to understand. If you want some publicly available free tools that you want to use, here's a course using R. Okay, so we have a collection of materials here that you can access. Projects, right? If you want to learn in greater details